I'm Sam Hudson.
Yep, good, go ahead.
I'm Sam Hudson.
And I'm Bryce Vaughn, and we'll be talking to you today about our presentation of the lethal and sublethal effects of pecan and peanut fungicides on natural enemies. And we were mentored by PhD candidate Pedro Toledo.
Pesticides have been used throughout the world for a very long time, and they are very crucial in agricultural success. Pesticides are designed to control or eliminate a pest in a biological setting. There are several different kinds of pesticides, ranging based on the target that they are designed to control or eliminate, such as herbicides, insecticides, fungicides, and many others. All of these are known for their potential to be a threat to non-target organisms and the environment as well, with overuse and just extended types of use. But of all these types of pesticides, fungicides are the most used throughout the world and most common on the market. Of the non-target organisms that pesticides in general can affect, one type of non-target organism that we wanted to look at was beneficial insects. And beneficial insects are important to the environment, especially in an agricultural setting, because they offer different niches, such as honeybees that are pollinators, lady beetles or ladybugs, as most people know them, by being natural predators, which control other insects that are pests, and other species of insects such as decomposers. So with natural enemies, they are a very important category of beneficial insect that it adds a factor to agricultural success. They take care of pest insects by eliminating and eating them as food, pretty much, and they're very important to the environment. But we chose to look at these beneficial insects because they are commonly exposed to fungicides in agricultural settings. These fungicides, most have been tested and have been deemed as not a threat to non-target organisms, but little has been done regarding the effects of fungicides on non-target organisms and natural enemies specifically, as compared to other pesticides. So in our experiment, we took a little deeper dive into the fungicides and their ability to be used in the field as being safe, like they're considered to be. We tested these fungicides on two species of beneficial insects, which were lady beetles, and we chose the species Hippodamia convergens, and green lacewings, which is Chrysophylla carnea. And we tested 6 commonly used fungicides in the pecan and peanut industry. And what we did was we picked these fungicides because, especially in Georgia and in other parts of the country and world, these fungicides are used and could potentially impact beneficial insects.
Now we'll get into the goals and objectives of this experiment, and the main goal was to determine the short-term and long-term effects, along with the lethal and sublethal effects of fungicides on lady beetles and lacewings. The objective was to quantify the mortality rate and of the adult lady beetles as they were affected by fungicide. Then we also wanted to quantify the egg emergence rate and peak emergence of lacewing eggs after their fungicide exposure. Then we also wanted to quantify larvae mortality and survivalship through their metamorphic phase of green lacewings after they were affected by fungicide. So let's get into the methods and how we created this study. So first we had to create a study design, and we were using a baseline study. It was a beginning test, so we were doing it in a laboratory as previously mentioned. So we decided to use petri dishes and filter paper to apply the treatment on. Next, after we got all of our design created, we collected the insects, but for that time, the experiment started was in the winter, and there was no bugs or leaves or anything for us to go out into the field and collect. So we ordered from Nature's Control in Phoenix, Washington, and as you can see, we got 500 live lady beetles and 2,500 green lacewing eggs. So after them came in, it was time to figure out what our treatments were. And the treatments were a control that was distilled water, a fungicide A that was a Last 400, a fungicide B that was Miravis tops, a fungicide C that was micronized sulfur, a fungicide D that was Bravo, a fungicide E that was K-Phyte, and a fungicide F that was Supertein 4L. And all of these fungicides are commonly used in both peanut and pecan industries. Next, we had to figure out our concentration rates so we could figure out how much of the actual fungicide we needed to put into the mix with the water. So we mixed 20 milliliters of water, and we got our concentration rates as shown here. And to get how much product we needed, we would take that 20 milliliters and multiply it by your concentration rate to get how much of the actual product needed to be mixed in with the water. Next, it was time to apply the treatments to the filter paper, and we had two different types of filter paper, and we had the larger size up at the top that you can see, and that was used for our lady beetle adults. And the reason for this was because we were able to put multiple in a single dish. We were able to put 5 in a single dish. So we had 5 ladybugs with 5 replicates per treatment, giving us 25 lady beetles. And we let after the application was sprayed, we let it sit for 30 to 45 minutes because of to get the dry residue effect. Because in the field, they have the ability to leave the plant or avoid the spray as it's coming down through the orchard. And then next, we went on to the eggs, and the eggs were in the smaller dish, and they would receive direct contact. And the reason for this is because an egg is not going to be able to move. They're going to get direct contact as the sprayer is going down to the orchard. And we had 8 replicates per treatment of that, and there was 15 to 25 eggs per the treatment. And then next, we had the larvae experiment, and it was done with the smaller filter paper. And the reason for this was because during the larval stage, they express cannibalistic behaviors, so that would not be good for our treatment if they're just eating each other. And they receive dry residue because whenever, like the adults, they're able to avoid the spray as it's going down through. They can hide underneath a branch or something like that. And then we had to add everything into it after everything was dried or after we sprayed them with the direct contact. Next, it was time to place the insects in a controlled room, or right here, as you can see, we had a controlled chamber, which was a growth chamber, and we were able to control temperature, the light that it was on for like simulating day and night. We were able to control the humidity and things like that, and we placed them in there for roughly around like 20 days, but most of the monitoring was done after 10 days for like the lady beetles and things like that. But the larvae, we kept them in there for their whole entire metamorphic phase. The monitoring was for every 12 hours for the first 5 days, and then after that we went to 24 hours until we were done collecting our data. And the average temperature was 25 degrees plus or minus 2 degrees Celsius, and the average relative humidity in the chamber was 60 plus or minus 10%.
With our data analysis, we had 3 tests, and we used a one-way ANOVA test for most of that with a alpha of 0.05. And if a statistical difference was detected while we were running our ANOVA tests, we utilized Donut's method as a way to compare these statistical differences back to the control specifically rather than the variables. So we could manage if there was a statistical difference specifically just from the control rather than each variable to one another. With our first test, we measured the lady beetle mortality, and after 5 days, here you can see on our graph, there was no statistical difference in between the control method and the mortality of lady beetles exposed to the fungicides, as indicated by the percent on the y-axis. And then after 10 days of monitoring, we had one fungicide that had a significant difference, which was our fungicide C, which is micronized sulfur. This is an organic fungicide that we used, because of our smaller sample size, there were two fungicides that were almost statistically different from our control, being fungicide B, Maravis top, and fungicide D being Bravo. If we would have had more time or more of a sample size, there's a possibility that these would have been very close to showing statistical differences. But with our sample size and the conditions that the lady beetles were in, we feel like this was a part of the reason that the data turned out the way that it did. But it was very interesting that we found a correlation between lady beetle mortality and micronized sulfur. There we found several studies that showed that micronized sulfur could have significant effects on the metabolic rates of non-target organisms via absorption of the sulfur once exposed to that fungicide. Additionally, we have also found a study that sulfur could have repelling or negative effects on predatory insects. So to see that there was a significant difference with the sulfur, it confirmed what we had researched. We did more research on lady beetle mortality and found there was a study from 1990 that indicated some pretty significant mortality with one of the fungicides that we ended up testing, which was the last 400. When we tested the last 400, we were expecting or wondering if it would line up to the research we found, and what we found was that there wasn't a significant mortality as compared to that study. This could have been influenced by several factors like environmental conditions, rates of fungicide. We used a rate off of a 50-gallon tank mix, and some of the rates used in the other study were more or less severe. And they had a larger sample size as well, which could have also impacted their results. With our egg emergence results of lacewings, this was part of the second test that we ran. On our y-axis of this graph, you can see the total percent of lacewing eggs that hatched. And what we found was that the complete percent of the total eggs hatched was not significantly different than the control. And then we dove into that a little bit further, but as a whole with the percentage of the eggs hatched, it was not statistically different.
So as we looked at the egg emergence, and there was no statistical difference, we thought, what if there was a difference in the time that they emerged? If they emerged earlier, at the same time, or later? And from this graph, we used a Peaks curve fit using a nonlinear regression function from sigma plot. And you can see in this graph that your fungicide E, your K-phyte that runs right here, and your fungicide F, which is your super team 4L, had a earlier emergence compared to your other treatments and control. It was within the 24 to 48 hour window compared to the other ones that was roughly started around 36 to 72 hours. So from this, we concluded that the two fungicides, K-phyte and super team 4L, caused the emergence to be earlier compared to the other treatments and controls. Next, the next test that we did was the lacewing larvae, and this was a test where we wanted to see if their metamorphic phase was changed by the applying the fungicides. So we started with the initial date that we started at, and we recorded until they became adults. So they went through all stages of the larval, all instar stages, then we went through the pupation period until they became adults. And on this y-axis, you can see the days until it took them to become adults, and on your x-axis, you have your treatment and control. And we had one significant difference that was fungicide E, which is your K-fight, and it actually reduced the number of days that it took for them to become an adult. Next, we wanted to look at if the fungicide treatment during the larval test through their metamorphic was if there was any change in the weight. And the reason for this was if the weight was to appear to be lower, then they wouldn't have as much reproductive capabilities, they wouldn't produce as many eggs and things like that. And on the y-axis, you can see the adult weight in milligrams, and on your x-axis, you have your controls and treatments. And from our statistical test, there was no significant difference in the weights. So it was stating that actually the fungicides were safe on the reproduction portion.
So with our lacewing data, we determined that there wasn't a statistical difference between the percentage of lacewing eggs hatched or a delay in the morphism of lacewing development. We found a study from 2019 that indicated that fungicides could possibly delay the morphism of predatory insects from the larval stage to the adult stage. And so when we compared our research and our findings, we found that our fungicides tested did not cause a delay. I will also say that what the insect was different. It was a wasp larvae, but we did look at the fungicides that were used and compare those to our results with our fungicides on the insects tested. So with our fungicide E, we saw that K-Fi seemed to increase the hatch rate of lacewing eggs and the larvae metamorphosis. And in addition to that, there wasn't a negative impact on the lacewing adults that we weighed from the morphing phase with the K-Fight. We don't know necessarily what caused the K-Fight to speed up the egg emergence rate, or if there was a positive effect on the hatch rate and success rate of lacewings due to this fungicide, because there would be more testing that would need to be done in order to say if it truly was a positive impact. We would have had to look at the adult life of these insects that were affected by the fungicide to see if they were able to reproduce or if their lifespan held out as it normally would without fungicide exposure.
So to wrap things up, most of the fungicides that we tested had a low overall toxicity to the lady beetles and lacewings. But we also did find that there was some sublethal effects, and we warrant that there needs to be further testing done with higher replicate numbers. Also, you could test this in a tree. In a lab setting still, it would still have to be in a tree because there's really no way that you can watch a single insect in the wild. You would have to put a dorm over like a sapling and do more testing and things like that. So just more testing needs to be done to confirm what we found. We would like to thank Melissa Thompson of the University of Georgia for helping order and assembling the materials whenever they arrived. We would also like to thank the University of Georgia Department of Entomology Tifton campus for the opportunity to work in the lab and conduct the experiment. We would also like to thank the Southern Research and Agricultural Education SARE and the University of Georgia for funding. Also a huge thank you to our man back there, Pedro Toledo, for mentoring us and helping us through this time. And just thank you for everybody that helped us through this time. Questions? Got bugs?
Questions?
Yes.
So for the study that found differences in results than you did briefly mention that they use different concentrations.
Mhm.
What you did not mention, and my question is, did they differ in the application type because you were dealing with residual
Yes.
rather than direct for the ladybugs anyway?
Yes, they were direct contact.
Okay.
So we tried to change that and give it more of a field experiment, like what they could be able to do in the field, they would be able to avoid it, like fly away or hide underneath the branch or something like that.
Okay.
So if you were to design an experiment, if you were going to take it to the next level, and you had unlimited resources, you could just design what you want to accomplish. Just pick one of them, either ladybug or lacewings. Just pick one of them, and how would you do this again?
With the lady beetles, I would say that a much larger sample size would be needed. In addition to that, I think that some more extremes could be thrown into play as far as concentration rates of fungicides and even application of fungicides. Although residual is more realistic in a field setting, to really see whether the immediate impacts of those fungicides could be lethal. I feel like it would be more advantageous to do a direct application with stronger rates to go ahead and build the basis on further testing, so time would be used more efficiently.
You could almost build it up to, all right, here's the threshold.
Yep.
This is the concentration, it's too much.
So do you think with the concentrations that you had now, you would have gotten different results if you had done a direct application aspect as well? Or do you think it had to be that higher dosage in the other study?
I would say probably the higher dosage, just because you're gonna the more the dose that you have, then the more lethal it can be.
Especially with the lady beetles, they were monitored for 10 days, and in that 10-day period, they were not fed. So even with that extreme environment, we still saw, it still took 10 days with our low sample size to see a statistical difference in the control versus one of the fungicides.
How much genetic diversity do you think you had in your samples? Like, you got them from one source. Do you know if there's a lot of progeny building where they make sure there's a mix, or is it just at their whim? Oh, well, she gave like 10,000 eggs. Let's give you all the eggs from that one individual. Do you know what the source of the insects were and whether that would affect things, or do you just expand on that, if you would?
We do not know if all the eggs came from one female, or if they came or what. So I mean, that would be something that would definitely need to be on another study, collecting them in the field, so then you have your true randomization, and it's in this area. Because I mean, as I mentioned, it's all the way in Washington, so you're not exactly getting what's in the field here.
So you said you get ladybugs to produce eggs?
Ours did not.
Yeah, ours were unsuccessful in reproduction, and we're not entirely sure why.
Needed some berry manilow.
Yeah. You give them a little wine or something.
Any other questions?